Kia ora koutou katoa. Nau mai, haere mai. Welcome. My name is Michael Kingsley. I'm the Interim Dean of Science here at Aotearoa, the University of Auckland. It's a great pleasure to welcome you all to this inaugural lecture of Professor Greg Holwell, a scientist whose curiosity of the natural world has taken him on a journey from the bushlands of rural Australia to the rainforests of Queensland and now to the lecture halls and field sites of Aotearoa. Along the way, Greg has studied kangaroos, platypus, praying mantis, and even spent time teaching high school science, a chapter that speaks to his deep commitment to sharing the wondrous world of the natural world. Since joining the University of Auckland, Greg has built a research program that's low-tech, low-budget, but high in creativity and impact, leading to fascinating findings in behavioural ecology. He's a passionate teacher and mentor, inspiring students in zoology, behavioural ecology, and entomology. Greg is a strong advocate for connecting with nature and for recognising the value of studying the underappreciated creatures that share our world. Tonight, he will take us on a journey through the stories invertebrates have told him, stories of evolution, adaptation, and wonder. Please join me in welcoming Professor Greg Holwell to the stage.
Wow, this is the best lecture attendance I've ever had, actually. This is impressive. So, tēnā koutou katoa. Ko Gary word te maunga, ko Wimmera te awa, ko deep lead te ngāhere, no Australia aho, ko Greg Holwell toko ingoa, no mai, haere mai. It is an absolute pleasure to see you all here today. I'm kind of overwhelmed by the number of people that have turned out. It's really special to see lots of people that I have known for a long time, people who I've known only recently, all coming along to have a listen tonight. So I hope I can make it entertaining for you. Particularly wonderful just to see a lot of my postgraduate students here, current students as well as past students who have, some of them travelled quite a long way, have flown up from Christchurch and Wellington, and it's really special and really meaningful to me that you've done that and made that effort. So I really appreciate that. And particularly to have my family here, to have Anne and Leo and Hazel here supporting me, and also that my mum and my dad and my sister Jodie all came over from Australia for this. It's making this a particularly special night for me. So thank you all. What I would like to do is really just kind of take you, I suppose, from the beginning of my journey and my fascination with nature and the natural world and natural history, which started at a very young age, right through to what I'm doing today. And the fact that excitement about the natural world and about studying natural history and making natural history an important part of the scientific endeavour in the natural sciences, it's still something that I feel really passionate about and what drives me in my work all the time. So I want to talk to you about that journey along the way. I also want to, I guess, share a bunch of interesting stories of things that have happened along the way, some of the research that I've done, but I particularly want to highlight a bunch of the research that my amazing postgrads have done, because they've really driven the majority of the research that I've been involved in. Some of those I'll highlight in a bit more detail, some I won't get a chance to get into because, as you'll see, I've had a lot of students. I don't have too much time to get through them all, but I will try to mention just a cherry pick a few highlights out of there for me. So my journey started in this town. This is Stawell in Western Victoria. It was a mining town, farming town, but also had a lot of bushland around it, and the amazing Grampians National Park in the background there, which was our playground and an endless source of wonder and fascination for us growing up. It's my family home growing up. A wonderful place to be, a wonderful place to grow up, lots of wonderful memories from there. And I was born in Stawell and grew up. I had my whole childhood in this home. I was pretty cute when I was a baby, but all babies are pretty cute. There I am with my beautiful mum. And I think right from the beginning I was a bit of a bird nerd. But I can thank my personal stylist, my sister Jodie, for that one, and for a range of other outfits that she stuck me in. But probably the key sort of memory of my childhood growing up in this area is just the amount of time spent out in nature and exploring the areas that I grew up in. So we would go camping a few times a year. We would spend a lot of time out at the lakes around the town, Lake Fynes and Lake Lonsdale, where we would, a couple of times a week during the summer period, Mum and Dad would bundle us in the car and station wagon and take us out there for a picnic or a swim, or we'd play cricket in the lake, which is a challenge. And we'd go yabbying. Those of you unfamiliar with the term yabbying, yabbying is putting out pots to catch freshwater crayfish from the lakes around that area, and we would get pretty big hauls. And there's a bunch of yabbies cooked up, my granny holding one of those. So a lot of time in those lakes. A lot of time out in the Grampians National Park. This spectacular scenery is just magnificent. If you haven't been there, I really encourage you to go if you're ever going to Victoria. A lot of time outdoors, and a lot of time on some pretty crazy adventures with my older brother Scott, who couldn't be here today, but I think he'll be watching the recording. His adventurous spirit really rubbed off on me, and we used to just head into the mountains and just go off track and see where we ended up. And it was sometimes a little risky, but a lot of fun. So this kind of time in nature was such a critical thing for me growing up, and it has continued to be, and that support that I've had from my family, from my brother and sister and my mum and dad, just in connecting with the natural world has just been a wonderful part of my life, right up until, here's a photo of us all only a month ago, back out at the Grampians all together, heading out there as we always do. We always gravitate back there, and it's a lovely place to go. So, as well as just a connection with being outside in nature and getting out into the wild places, I just always had just a love of animals. I wasn't somebody who had pets or who brought animals home, but I was just fascinated with them. I just loved to go and see them in the zoo, and I loved books about animals and about nature, and my parents and my brother and sister would buy me books like these ones here about, just that would just enthrall me, and every year I'd get more books, and my mum would read me these nature books. My sister read to me a lot, and I think I got a real love of reading and of writing from my sister through that, and just that engagement not only with being in nature but also in exploring what other people were finding out about nature and about the animal world. And that was always something that got me really excited. So, as I said, I also loved to read. I also loved to write. Really enjoyed writing stories, and I've continued to love writing to this day. Some of you I've taught in postgraduate writing workshops, and I still enjoy the process of writing. It's a wonderful thing. But I did come across this old kind of journal from school when I was visiting Mum and Dad. And so I have enjoyed writing for a while. Here we go. Today I got up, got dressed, had breakfast, came to school, and now I'm sitting at my table, happy as can be, writing my diary. What a joyful existence. And it contrasts a little to some other entries, such as this one, on the 10th of December. I'm tough. I perhaps didn't have quite so much to say on that day for my journal. This one I quite like, Monday the 14th of October, I might see Back to the Future. 15th of October, I didn't see Back to the Future. So a bit of an anticlimax, I suppose, there, but just a nice little snapshot. And if you're familiar with the Back to the Future movies, 1985, this was 1985. This was critical time period in the Back to the Future movies. So, basically exactly 40 years ago. So there I was as a 9-year-old, and I really enjoyed writing. But perhaps most prophetic in this journal was this entry on the 6th of November, which is, I want to be a zoologist and study animals. So it's kind of interesting, and I do really clearly remember that being at that age and kind of discovering, just being passionate about watching shows about animals on TV, reading books about animals, getting out in nature, and then to find out that it could be a job, that going out and learning about animals and discovering things about animals could be a possible thing that you might do with your life. And that really blew me away and became really sort of my passion from that point onwards. So I pursued that from that sort of time until the present day. And it was pretty much a straight line, although there's always a slight detour in one's life. And for me, it was when this happened. So at about the ages of 14 or 15, I went through this phase where zoologist sort of took a back seat. Was lead guitarist in a metal band was the dream. Amazing time of my life. My good friends Macca, Matt and Mick, who I played in this band with, continued to play in a band right through my teenage years, and who then went on to professional music careers. So it was a really exciting and passionate time of our lives. Really, really wonderful thing to experience. But I think underneath it all, I was always still hoping to pursue this career as a zoologist. Love my music, still listen to music, still play the guitar, but zoology has been a little easier to find a path in terms of my career. So back to studying zoology. So to continue that, I needed to go down to uni. And again, my family was so supportive in helping me to get there to go to uni. As I know it was a big financial burden, and I know Mum had to keep on working to help me to get there, and I'll be forever grateful for that. And going down to Melbourne to study wasn't easy. I had some great friends that came down from Stawell at the same time, and were a great support network there. But I didn't find university study easy after high school. I went from being an A grade student down to being more of a B and C grade student through my first couple of years at uni. And it really was only in my third year that I started doing subjects that caught my excitement again. Natural history-based subjects like biology of marsupials and animal behaviour and conservation biology, and the sorts of courses that got me excited about nature again. So eventually I got to the end of that degree, majored in zoology and did some botany as well, but finally got there and decided to continue on and do an honours degree. And for that I worked with Graham Colson, who was my supervisor at the top left there, and I did my honours project, which was kind of more of equivalent like a master's project here on kangaroo and wallaby ecology in fragmented forests in Western Victoria. So I could go back home to my home area, spend a lot of time there, do my field work there, and get out chasing kangaroos down in my 1976 Ford Cortina and fanging around the bush, and also doing a lot of transects in really hot weather. I remember my dad out helping me doing a lot of these transects, and the last day was on my 22nd birthday. It was 45 degrees Celsius and we were doing this transect through the bush. It was intense, but a really great experience. And interestingly for me, I think it wasn't so much just the project itself, but the time that I got to spend out in the bush that really, I learned more just from immersing myself in natural history through doing that. And I would love buying field guides and taking them out and figuring out what plants I was seeing or what birds I was hearing or what reptiles I would see. And this particular book, Tracks, Scats and Other Traces, it's a brilliant book because it's actually hard to see most of Australia's mammals. So to get out there into the bush and see some scratching on the ground or some pile of poo, you got this field guide out and you'd sit down and try to figure out what had done the poo or what had scratched that hole. So I found that really incredibly rewarding to immerse myself and learn a lot about natural history myself just through time in the bush. as I said, I wasn't somebody who tended to bring animals home to kind of keep them as pets, but what things that I would find I would bring home. I loved bringing home eagle feathers and nests, and I found a skull once and brought it home and figured out it was a koala skull in a place where there really shouldn't have been koala. Really kind of interested in that sort of thing. I've got a picture here of a little feather-tailed glider, lovely little species of possum, only about the size of a mouse, but they have a patagium so they can fly from one tree to another. Gorgeous little things. This is a lovely photo that my dad took. But it's here to remind me that not that long ago, Mum gave me a call and said, Greg, there's this little furry ball in a bag at the bottom of the freezer. Looks like maybe some kind of possum or something. And I said, oh yeah, that'll be that. You know, it was a feather-tailed glider that I had found dead on the road at my sister's farm and taken it home, put it in the freezer and forgotten about it. And about 20 years later, Mum dug it out. So this was the kind of thing that was happening all the time. So sorry to Mum and Dad for that. Another incident, dead goanna, found on the side of the road again, screeched to a halt, had to have that, pulled it off the side of the road and threw it in the boot of the car, and unfortunately once again forgot about it. And about 2 weeks later it was, what's this? I can tell you, if you really hate somebody, stick a dead goanna in the boot of their car somewhere. One of the worst smells I've ever experienced in my life. But that didn't stop me then pulling it out of the boot and skinning it and sticking the skin on a board and forcing Dad to put up with that hanging in the garage. And so this sort of stuff I just really loved, and just that immersement in nature and the natural world and finding out what things were was what really got me excited. So after my honours degree, I was also pretty fortunate to get a job, and back home in Western Victoria, giving me the opportunity to stay in Stawell and this time get out into our river systems, the Wimmera River, and work on platypus. So here I was employed to radio track some transmitter-fitted platypus through the Wimmera River. Really degraded river system. It runs through farmland. It's not pristine at all, not the kind of place you would expect to see a platypus. So that was really interesting to kind of figure out how they were using such a heavily modified environment. And again, there's Dad with me in the river. Dad would regularly come out and help me throughout all sorts of phases of my career, and that's something I always really appreciate, Dad. So that was a fun year. So after this I moved to Sydney and I did a teaching diploma at Macquarie University, and I taught at a high school for a year. I think always along the way I had this view that I was going to kind of get some skills and experience in teaching to kind of match with my research experience in a hope of kind of pursuing an academic career. And I think that really has stood me in good stead to follow that path. But eventually I returned back to that point of wanting to follow that academic career and knew that I needed to do a PhD. So this was when I met Mariella Herberstein, who had just set up her lab there at Macquarie University. Instantly we really clicked. I could tell that she was going to be a great supervisor for me, very supportive, but also somebody who gave me a lot of freedom to take the project wherever I wanted to take things. And we had lots of really great discussions. She set me up joined in a few other research projects that were happening. She organised for me to get a bunch of TA work to start paying the bills, and really, really great supervisor. So working with Mariella really kind of set me up. And one of those early conversations after I'd kind of got underway was her suggesting, well, would you be interested in working on praying mantises? Because I think their behaviour would be really, really interesting. And I was kind of blown away at the thought, and I went and explored a little bit, and then I was even more blown away to discover how little we knew about praying mantises, which seemed to me one of the coolest groups of insects out there. So I was hooked, and I really wanted to go for it. So we started looking at praying mantises around Sydney, started collecting them, observing the behaviour in the wild, and we started seeing some of these sorts of things. So let me just see if I can get this. There we go. So there's a male on the left, a female on the right there, of Pseudomantis albofimbriata. The male's flown in there. The tension is palpable. There we go. And no wonder the tension was palpable, because this is sort of the fate of many male praying mantises for this species and for a number of other species. A lot of mantises show sexual cannibalism, which is a really unusual and really amazing and fascinating behaviour, whereby females will eat males, often before they get to have the chance to mate. But regularly they can continue to mate despite having lost their head or part of them having been eaten. So a really extraordinary behaviour, common in mantises, common in spiders. That was the shared thing. Mariella had worked a lot with spiders, and that was her sort of thought with why not kind of go in and think about working on mantises. So thinking about this kind of mating interactions, really ferocious interactions, really fascinating, trying to get to the bottom of what the adaptive significance might be for this sort of behaviour. Led to some really productive work. So over the years, this has really been productive through a lot of collaborations, and a lot of students have generated some really interesting work, including a paper there by Leilani, who's out there somewhere at the top. So lots of cool stuff on cannibalism and mantids. But this was kind of all a side project. I actually didn't work on cannibalistic mantids for my PhD. I sort of started to work on those sorts of topics with side projects and get students involved in them during my PhD and my postdoc, and have continued on to that point. But for my PhD, I fell in love with these little guys. This is the praying mantis genus Schilfina, and I collected them the first time I went on a field trip up the Queensland coast, and I found them in really good numbers. So, you know, at a particular location, I might find 100 in a day. They live on tree trunks, always on tree trunks. They're small, only a few centimetres long, long-legged, incredibly fast. They run up and down tree trunks incredibly fast, catching their prey actively rather than being sit-and-wait predators like most mantises. And so they seem to me to be a really unusual group, and a group worth studying. So having them in good numbers, having them being a group that nobody had studied before was really exciting to me, and Mariella supported me to kind of take them in whatever direction things were going to go. But the problem sometimes with studying something that hasn't been studied before is that they don't even have names. So I had to fairly quickly also become a taxonomist. I had no training in that, but I had a student, Scott Ginn, who had done a lot of taxonomy, and met some wonderful taxonomists, John Balderson and David Rentz, who supported me in that. And so first task, figure out what the new species were going to be, describe those new species, give them some names. And to do taxonomy meant that I had to learn a lot of morphology and anatomy, so that really immersed me in that too. So starting out with more of an interest in ecology and behaviour, I also had to pivot and learn a lot of morphology and a lot of taxonomy as well. But it was a really rewarding thing to do. But what I really loved was getting out into the field, and this was the kind of silhouette that I was looking for when I was out there, Schilfina up against a tree trunk. When you see them on the side there, it really sticks out, and this image was just absolutely burnt into my brain as a key to find them. Once I was driving along on the road with Anne in Queensland, driving at 60 or 80 along the road, and I pulled over and said, I just saw a mantis. And this is a true story. I got out and there, sure enough, there was a mantis on the tree. I'd seen that silhouette even driving that fast past the tree. So really loved being out there in the field and doing some ecology and some behaviour as well. And it took me to some pretty spectacular places. So I took every opportunity to get up there and do fieldwork in this wonderful place, Mission Beach, and we've continued to do fieldwork up around that area. It's a glorious part of Australia. And perhaps one of the most interesting things that happened whilst doing fieldwork in Mission Beach was in 2006, the biggest cyclone ever to hit Australia, headed straight for Mission Beach when I was there with 3 of my honours students working on Schilfina. We thought probably a good idea to get out of there and talk to a lot of the locals about what usually happens when cyclones hit, and do they tend to head north, do they tend to head south, because it's normally what happens with a cyclone, they follow the trajectory of the coastline. It wasn't very helpful. They sort of said it's kind of 50-50, so I thought, all right, I packed the students up in the car and we just drove straight inland. So we headed across into outback Queensland, straight to a town called Georgetown, about five hours inland. But Cyclone Larry, biggest cyclone ever to hit Queensland, it decided to just go straight across right over the top of us. It had, to be honest, it had calmed down a lot by the time it got to Georgetown, but it was still pretty exciting to bunker down in a motel with a cyclone going over the top of you. So pretty exciting times, but it kind of taught me a nice lesson because one of my great students, James O'Hanlon, and I followed up on that, and we continued to monitor the Schulfina populations there at Mission Beach post-cyclone. And we got a nice little paper about the cyclone responses, or the population responses of these mantises to cyclones. And as far as I know, it's the only study to ever show an invertebrate response to cyclones. So kind of turned got a silk purse out of a sow's ear with that one. Another thing about being there at Macquarie Uni was some great friends and colleagues that I met, some wonderful people who have continued on into great academic careers as well. And these are all, this is an incredibly blurry photo, but these were all wonderful, wonderful friends that I made during that time at Macquarie University. And working on Schulfina also ended up being pretty productive. So during my PhD and postdoc, we got a lot of work out there, a wide range of topics. So that was the beautiful thing about having a totally unstudied group, is we could take it and we could find out some ecology, we could do some morphology, we could do a bit of molecular ecology, we could do some behaviour and study all sorts of things. And I think it's something that I'm incredibly proud of, to be able to take a group of animals that is totally unknown, give them names in the first place, and then take the knowledge further and really find out lots of really fascinating and exciting things about those mantises. So, being there at Macquarie, got a lot of interesting research done and really exciting stuff. But probably definitely the most important thing to happen to me whilst I was there in Sydney was to meet this wonderful woman. Here's Anne and I. We're looking a whole lot younger there, aren't we? That's at New Bronte Beach, where I lived in Sydney, and we worked in the same lab, did our PhDs together, and have continued on our lives together. There we are, a little bit more like we look like now. Having Anne in my life through that time, she's the love of my life, and she's continued to just inspire me, and the support and love that she's given me has really kind of helped me do what I do today in every way. So thank you, Anne. This is a nice sort of special place to think back to as well, because I managed to take Anne here a couple of years ago. This is Gregory National Park, which is right in the middle of the Northern Territory in Australia, and I really wanted to take her back here because I wanted her to meet Schulfina and charlottae, which was the species of Schulfina that I named after Anne. And so she was able to finally meet Schulfina and charlottae, which was lovely. And I have to say that as a taxonomist, this is about the most romantic thing you can do. So it was a nice thing to be able to do that. Thank you. And following on from Anne, now within our lives we have the wonderful Hazel and Leo. And they're our Kiwis that we've raised since moving here to Aotearoa. And they continue to bring us joy and happiness in our lives every day. And they also continue to inspire me to do what I do, because they love joining me when we get outside and go on adventures. And we do lots of great stuff together. So thanks, Leo and Hazel, as well. So that kind of brings us to arriving here in Tamaki Makaurau. So moving over to Aotearoa was a bit of a surprise to find ourselves offered jobs here at UOA. So thank you, UOA, thank you, SBS, for employing us both to come over here. And we very quickly kind of settled in really nicely and really loved our time and continue to love our time here in New Zealand. It's a great place to be. So being here at UOA, I had kind of already had a reasonable amount of experience with sort of teaching and supervision and research, so I felt reasonably comfortable kind of stepping into the role. But moving to a new country, not knowing anybody, it's always hard. And so I had lots of wonderful colleagues, and many of you are out here in the audience today, and I want to really thank all of you for being here today, but just for that welcome and that support being here, the professional staff, the academic staff, the postgraduate students, the undergraduate students. It was really just a really great welcome, and continues to be a really great environment to work with. I'm not going to name lots of people in particular because I know I'd unfortunately forget people, so there's too many people to name here, but I did want to mention a couple of people that were really important in my arriving here. I remember meeting Mike Walker within SBS, and really inspired by his teaching, really inspired by his research, and mostly inspired by the fact that I could sit with him in the tea room and have a cup of tea and talk about the cricket and talk about family and talk about life. And he really kind of humanised and normalised academic life for me, and that was really, really critical for me at the time. But also Mark Halber, who was in SBS, and he was really welcoming to Anne and I, took us out on lots of dinners. And Mark was somebody who would regularly come into my office and sit down and have a chat, and just to say, you're doing a great job. And that was just really lovely. And so I really want to thank Mark for that, because it was really critical for me at the time. So I also want to pay a big thank you to colleagues out at the New Zealand Arthropod collection. So within pretty much a month or so of being in Auckland, I'd been contacted by Rich and Robert and Darren and Thomas, who were all like, oh, we work on these insects, and you've seen you work on those insects, and isn't that cool? And, you know, we should work together. And, you know, that's what taxonomists and naturalists do. And it was a really welcoming thing. And I continue to collaborate with these guys, along with Aaron Harmon, my mate from Australia, who's now working out there as well. So really strong connections that I've continued to maintain there with the folks at New Zealand Arthropod Collection, and they began right from the get-go here in Auckland. And here's my wall of fame for you guys. Most of you are up the back there. So this is my 30 PhD students that I've supervised. And every one of them has taught me some really, really valuable and important lessons. And the experiences that I've had getting to know them over the course of, 3 or 4 or 5 years within those projects has been probably the most special part of this career. Many of them are, still friends and colleagues today. And each of them did really exciting projects, took things in their own directions, and did really stellar work. And they've really pretty much propped up my whole research career through their hard work and their creativity and their talent. So a really big thank you to you all, particularly those who have been able to come along today, especially if you've come from some distance to get here. From here, I guess I just want to share just a handful of stories, just a few stories of some of the research that I've been lucky enough to be involved with, all student-driven. All of the examples that I think I'm going to talk about have been, as Mike introduced at the beginning, pretty low budget, pretty low tech, but incredibly high on the creativity, incredibly hypothesis-driven research that has taken natural history to make major impacts in various fields within behavioural ecology. And that's something I'm so proud of all of those students in being able to do, because it's something I'm passionate about. So I could have picked all sorts of different examples from the students' work, but I've just picked a handful to kind of explore in a little bit of detail. And the first one I wanted to explore with you all, because it's such a lovely story, is that of the orchid mantis, which is a Southeast Asian species of mantis. And started thinking about these because James, who had done a master's on Schilfina with me, wanted to continue on with a PhD with Mariella and with myself, and wanted to, started thinking about the idea of working on the orchid mantis. And we thought, well, we don't know how achievable this might be, but let's give it a shot, let's try this out. So Mariella and James and I flew to Malaysia and got out there into the rainforest to see if we could find some. And immediately, just the diversity in the tropics like this are just so spectacular. Just unbelievable. It's like a kid in a candy store, being an entomologist going out into the tropics like this. So within the first day of being there, I'd seen this huge, most amazing millipede we'd ever seen. I saw the most spectacular spider that I'd ever seen, and encountered praying mantises that were just mind-blowing and really spectacular as well. So just within that time, amazing diversity, amazing animals out there. But we didn't find any orchid mantises to start with. It took us some time. We searched around, had a good look around to see what we could find, but to no avail. But after a while, we decided, well, let's kind of head further afield. And we decided to drive up into the highlands and had to get up in the altitude a little bit and drive through some pretty misty mountains. The first time we went up there, it was a bizarre experience because we emerged out of the mist and drove under a roller coaster. into a casino and ended up having a robotic gondola ride in the casino. And there's probably some sort of lesson to be learned from all of this, but I really don't know what that could be. Frankly remain entirely confused by the whole experience. But I'm still very glad that we did it, because as we drove on our way back down from the Genting Highlands, we stopped to have a bit of a look in a little bit of bush, and there we were, a spectacular orchid mantis. We found one, and then we subsequently found a few more. And they really just are exquisite. They range in colours from white through to pink, and just these petal-like legs. They're just beautiful, beautiful creatures. And really fascinatingly, when we first found them, all of them were just out on their own, sitting on the foliage like that. We were very much expecting to find them on flowers. All the nature documentaries had them sitting on flowers. Photos on the internet had them all sitting on flowers, and realised that they're all just rigged. Basically, orchid mantises didn't seem to ever be found on the flowers like that. So we started really thinking about what might be going on, and James really came up with this wonderful idea and hypothesis that the orchid mantises weren't trying to camouflage in amongst flowers, they were being the flower. They were mimicking the flower in order to lure in pollinating insects to feed on. Because there are a lot of pollinating insects out there in the tropics that are going to and checking out all the flowers that are around to get nectar and to get pollen. So this led us to a very simple field experiment that we set up on the fly, tried it out there, and I love talking about this. I love sharing this experiment with undergraduates. Probably all of you heard me talk about this in classes in Biosci 207, but I love it because it's such a simple experiment. It's taken a simple natural history observation, turned it into a hypothesis that could be tested with in a simple way to get some interesting results. The equipment list for this experiment was 3 sticks. And we would set up those 3 sticks in a triangle. We would put an orchid mantis on one stick, a flower on another stick, and we would leave the 3rd stick empty. And then we would sit and we would watch to see what happened. We blew the budget out a little bit and we got more sticks. And we set up groups of 3 sticks in different parts of the rainforest, and we all set up in different places and watched these triangles of sticks to see whether pollinators might be coming to check them out. And indeed they did. So not many insects went and sat on the bare stick. Quite a lot of pollinators came and hovered around in front of the flower or landed on the flower, but even more insects would hover around right in front of the face of the orchid mantis, and many of those would get nabbed. And here is a nice little video of that in action. Oh, here we go. It's catching up. So this is what the orchid mantis is all about. It's mimicking, using aggressive mimicry, pretending to be a flower, catch pollinating insects as they come in to try and when they think they're going for a flower like that. So really exciting stuff, and I love the story there, and it's a simple one, but it really shows how you can just take some simple natural history observations, do an experiment on the fly, end up getting published in a pretty good journal, the American Naturalist, and leading on to a whole lot of other really cool stuff that James went on and did with his PhD on orchid mantises. So I also continued and got really excited about working on mating systems of insects, and particularly the scramble competition mating system. It was something that I was interested in from early on, looking at mantises in Australia, but that continued on here, and a bunch of the postgrads worked on that, firstly with Shelley, who's here. Shelley chasing stick insects around out at Aotearoa, following them and looking at their interactions and their mating system out there. Bex, who's here, continued on with her PhD, part of that work looking at the mating system of these kelp flies on beaches, a great excuse to hang out on the beaches for your PhD, and do some really wonderful research on kelp flies. And then Neil, more recently, who's also here, and his hoo-hoo beetle work, very multidisciplinary PhD, but some of Neil's work was looking at scramble competition. I think this might work, let's give it a try. Here's Neil's. Here's where the technology gets pretty high tech. Neil built amazing contraptions out of Lego and electronics to build this flight mill so that hoo-hoo beetles would fly around and we could see how far, how fast, and how long these hoo-hoo beetles would fly for, and look at this in the context of scramble competition. So really cool stuff there. So also continued on with my research on the evolution and the comparative morphology of reproductive structures. It's been a really fascinating area to develop, and particularly early on after I arrived here. Did some work with John Evans at University of Western Australia on guppies. Brief foray into some vertebrates again, and also managed to get some funding success with a Marsden, and that funded Rebecca's PhD, who's here as well, and we were able to look at really spectacular degrees of diversification in the reproductive structures in these beautiful little lichen tuft moths. Camouflage has also been something that I've really enjoyed developing as a research interest and as a research kind of strength. Started off, I think, with Cass's work on lichen moths, and Cass did amazing stuff where she built hundreds of artificial models of these lichen moths, deployed them out into the bush to look at predation attacks by birds in the field. She was able to manipulate those models in different ways to see what was important for camouflage, and discovered really fascinating things like symmetrical wing patterns lead to higher predation than asymmetrical wing patterns, and that helped to explain why this group of moths have asymmetry in their patterns, which is incredibly rare. A really lovely finding there. Morgan continued working on the topic of camouflage, looking at stick insects. Morgan found some really exciting things about stick insects, such as that not only do they look and act a little bit like a stick, but they can even adjust their posture so that the angle that they hang from a branch matches the angles of the surrounding sticks that branch off from those trees. Really fascinating camouflage work. And then more recently with another Marsden fund with my wonderful colleagues Rich and Thomas, and our wonderful PhD students Kelly and Kyle, who did some really wonderful work here on these debris cloaking beetles, and found some really exciting stuff as well. So camouflage turned out to be a really exciting field to get into as well. So animal weaponry was another area that I got really excited about fairly early on, and a couple of branches of that was on some Marsden-funded research on these harvestmen. PhD student Erin Powell did some great work on harvestmen and their comparative morphology and these ridiculously exaggerated weapons that they possess. Their jaws weigh more than the rest of their body put together. Outrageous and really interesting things. And then Leilani developed and drove some incredible work on the Cambridgea sheepweb spiders, putting them into a really lovely comparative context, looking at allometry, looking at the evolutionary divergence of these amazing and complex weapons that you see in these sheepweb spiders. And Josie McCambridge did her master's on that group as well. So really exciting groups to study in terms of weaponry. But the study of weapons really kind of started with Chrissy when she joined the lab, and she was my first PhD student here at UOA, and started your PhD kind of not with really much of an idea of exactly what you were going to do for the PhD. We just started exploring all sorts of different ideas, and giraffe weevils were one of the groups that we got excited about early on, and decided to turn into a PhD. And thanks to John Staniland, pictured here out at Matuku Reserve, from his support and help in really getting there to work on giraffe weevil populations close to Auckland. That really made the whole project possible. And here's a great clip from Chrissy showing here's a pair of giraffe weevils, and another interloper has just turned up. They're all colour-coded so that Chrissy could identify who is who in the population. You can see the female's totally uninterested in what's going on there. She's off, but the males really want to fight it out there. Oh, and off he goes. Gets thrown off the tree. So this is the real context there with studying animal weaponry. The males are using them in these contests with one another to try to outcompete with one another to increase the possibility of mating opportunities. And certainly, you know, the initial question, why the long face? I tried to convince Chrissy to use this as a title for a paper. She didn't want to have a bar of it, but I just think it's brilliant. You know, giraffe weevil walks into a bar. So the data that we got on giraffe weevils really sort of set things up with thinking about weapon evolution. And we really discovered what's shown in this graph here. If a particular giraffe weevil has a longer rostrum than its rival, it's more likely to win the fight. If it's got a shorter rostrum than its rival, it's less likely to win the fight. And that was a really clear pattern. And it's something that carried on with the other species that we've looked at as well. But then Bex came to do a master's and continued on and did enormous numbers of hours out there in the field, monitoring populations over months, individuals. Huge amount of data has gone into this, and effectively what Bex was able to show was that this winning of fights also correlated with higher mating success for larger individuals with longer rostra. This was the case for males and females, and the larger giraffe weevils also continued to have longer lifespans as well. So it seems to be a win-win situation for these big giraffe weevils. The cave wētā are another really wonderful group that I really enjoyed, again partly for the location. Was able to get to know Anne and Alistair Stubbs from being introduced by Carol Bannock, who introduced me to the Stubbs, and was able to get down there and start exploring their amazing property at Waitomo. A lot of bush and a lot of cave systems, and we've all spent a lot of time down there taking the kids to explore those caves. And you'll see these cave wētā up in the top right there, covering the ceilings of the caves in these limestone systems. Really, really amazing group. And Murray came along, pretty interested in working on a project here. Murray Fee, he set up a whole bunch of infrared trail cams in these caves, looking up at the ceiling of the caves to follow these populations. And they're incredibly chaotic. And as well as seeing the way that the legs of the male cave wētā were being used as weapons, he also noticed that they were being used to effectively kind of deflect disturbances away from females. So you can see in the little clip here, a spider's crawling through this population, and it's scattering everything that it goes near, but there's a big male who's staying put, and he's managing to stop that female from being disturbed by the spider. And this happens incredibly frequently. So Murray tracked the paths of disturbances moving around through that population, many hours of footage of individual female cave wētā to show how often each one of these dotted lines represents a spider or another cave wētā just bumping into one of these cave wētā, how often they're getting disturbed. But when they've got a male guarded around that female, very few of those disturbances hit the females there. So a really, really fascinating finding. Effectively demonstrated that guarded females received far fewer disturbances than unguarded ones. Males with longer legs were more successful at being able to guard females in this kind of way. And the longer that males could guard those females, the more times they got to mate with those females. So a really lovely, once again, taking just a pure natural history observation of something going on, coming up with a hypothesis, thinking about a way to test that in the field and get some really interesting data. So all this work on weapon evolution and animal weaponry has also been really productive and led to lots of great papers from all my wonderful students who have driven all of this work. So lastly, what I really want to kind of talk about is once again sort of thinking about natural history, but thinking about it in a teaching context. Teaching is such a big part of my role here, and I see it as just as important as the research that I do. And I have loved incorporating natural history into the way that I teach. A lot of that has involved things like running an insect collection, and this was set up by Jacqueline, I think is here, who for BioSci 320. Still such an incredible assignment for students to actually get hands-on connection with insects to develop this collection. And I've done a lot of reading, a lot of thinking about object-based learning and the way that interacting with objects, whether they're specimens or live animals, can just enhance learning opportunities for students. It's been really critical. And so that has allowed me to kind of tap into this wonderful resource we have, the McGregor Zoology Museum, and a real hats off to Dave Selden for his leadership in curating this collection. I get students in there whenever I can. It's such a wonderful resource. Being able to get students in there to do assignments where they make observations of features of animals in the museum. They come up with hypotheses to explain those observations. They then follow through to think about how they might test those hypotheses. What research could they do? And they develop research proposals based on those ideas. And I think this is a really exciting way to get students engaged in natural history, but also give them those real skills of thinking about nature scientifically, thinking about hypotheses, thinking about how you might test them. And I think it's a really critical part of a science degree. One of the teaching activities that I've done that I'm most proud of, I think, came during a time of most challenge. It was during lockdowns and COVID, when our entomology class wasn't really able to get out of the house. So we set up a little project where students would take photos of the moths that came to their windows of their houses as they weren't able to leave their homes, post those photos onto the Canvas site, and other students would try to identify what they were. And this was really successful, led to really huge 100% participation in the class during a time that was known for such low levels of participation and engagement. We identified 79 species of moths amongst us all over a few months. And it also, thanks to Julie McIntosh's recommendation, led to a nice little publication about online entomology activities that we might be able to do. So the last point that I want to make was that last year at the ISBE, the big International Behavioural Ecology Conference, John Endler gave the Hamilton lecture. He's a behavioural ecologist, evolutionary biologist that I greatly admire. He's been prolific in the work that he's done. And he gave a lecture and subsequently wrote this up as an article. And I just want to highlight these last points here, because he's made the point that we need to make a lot more use of natural history. Natural history is the ultimate source of all hypotheses, so neglecting it could result in stagnation of our field. We need to think more about the origin of good questions and testable hypotheses. Good science is not just a matter of hypothesis design, but also how we generate the hypotheses in the first instance. And so to hear that from one of these leaders in the field just gave me great heart and great encouragement. And it also once more made me feel so incredibly proud of my postgraduates that are doing exactly what it is that John Endler was asking us to do in our field, to drive our field forward. All of you have made discoveries that have fed into the field of behavioural ecology, starting from the starting point of natural history. And I think this is a really not something to be taken lightly. You should really be proud of yourselves for that. I also take heart because it means that I can continue to get out there into nature, and all these interesting critters that are out just around Auckland that I find every time I get out into the bush are all ripe for more study, more natural history, continuing on into more behavioural ecology. And with the continued support of my family, getting out on a lot of these adventures to explore that natural history, I know that I'm going to have many more years of being able to do this ahead of me. So with that, I want to thank you all for being here today.